Ronald, Ronald, hey,
Morning, morning, Johnson.
morning, morning. Sorry for early start, right? But I wanted to catch everyone before they get on their day.
Yeah, thank you for arranging this. You've got a very interesting background that looks so real.
I don't know if this is real.
Where are you? In Shenzhen?
No. I'm at home in Gold Coast. But I,
Got
I just thought I wouldn't disturb my kid sleeping, so came downstairs.
got it.
Yeah, you thought it was AI generated background. Yeah. But anyway, I'll tell you the sequence of the call later, so you might need some slides, right? The first slide is I think I'm going to start by asking your team's background, right? Introduce your team's three founders and the NIH funding, okay? Then the second part is actually on the competitive field. How are we different from other AAV gene therapies? That means the intercalary, right? Hello, morning, Stephen.
Hey, hey, good morning, Johnson. Hello.
Hey, morning, guys. Hey, anyway，你先放在 mute 上先，好不好？我跟 教授 run through 那个slide 先，okay? So Ronald, so the first slide, right, that we wanted to talk about is actually the difference between the other players, right, and us and us bio, right? Then the other one is on the dosing slides, right? So how are we exactly similar with us bio and very different from Rocket, very different from Celadon, right? Yeah. And then the third part is actually on the mini hut, right? Novo hut. How does that actually help guide our dosing to get the right balance between efficacy and safety? So actually, these three parts should take about 30 minutes, right? And we still don't go into data. Or then at 9:25, right, we go into the low dose data, how to interpret it, right? And then when Professor Hajjar comes on, we go on to the high dose data, the new stuff I haven't even seen about, right? Yeah. So roughly those are a few slides.
Okay, all right, sounds good.
Yeah.
Uh, yeah,
Give
so
them a minute.
so should I start now? So let me
Oh no.
share
One
my
minute.
screen.
I'm just checking if everyone is I see a J and C. Is that Jacob and Charlene? I'm hoping that is right. And then I don't know whose iPhone. um I think Susie Shinzo is on. I'm gonna just remind Susie. Hold on, wait.
Can you see my screen?
Yes, we can.
Just let me know whenever
Yep,
when
yep.
you
1 minute.
guys are ready. So Johnson, did you say that you want
Yes,
Roger to join Professor Hajjan to join at 9:25? Because
nine
I
thirty. No,
I
no, no. It's okay. Okay. I'm just saying that we can lead into it at 9:25 on the low dose data. Yeah. Then we go into the discussion about the high dose data, right? Which is new, right?
because I
Yeah.
told him to join
On
at 9:30. Let's see.
just make sure everyone is here. Yeah. Hi. May I check who's the iPhone? iPhone on the list.
Hey, Jason, I think it's me, Sunny.
Oh, Sunny. Hey, Sunny，你
嘿，
好。
嘿，嘿，你好，
OK，
你好，
啊，
yeah，
不要紧。OK，I think
嗯，
let's get started，因为Susie 让我record 这个 call。好了，那行。OK，yeah. So Professor, please go ahead. Please introduce yourself and your co-founder, right? And then how do you guys manage to win that much in NIH funding, right? Please go ahead.
Thank you, Johnson. So nice to meet everyone. So thanks for joining on a Saturday morning. So I'm Ronald Lee. I'm one of the co-founders, three co-founders of the company. So Medera is a clinical stage, Boston-based company founded about 10 years ago, initially as an academic spin out of Harvard and Johns Hopkins, where our original technologies were first developed, basically by the three co-founders. As I said, I'm Ronald Lee. I'm functioning as the CEO of the group. And as a quick introduction, I was a professor I started some of the work that I'm going to be telling you about 25 years ago for me, and 30 years ago for Roger that you're gonna be meeting with very soon. And we started the work, and the three of us came from the same pedigree: Johns Hopkins, biomedical engineering, cardiology, moving on to Harvard. And about 7 years ago, we decided to become, for me to become the full-time CEO. I've been running the group ever since. So my specialty has to do with human heart engineering. And Roger is my more senior colleague and about 10 years older than me. And we've been working together for over 25 years on the very same topic. And he's going to be joining in a few minutes. And he also came from Johns Hopkins, moving on to Harvard. And currently, he's also the so he's president and chief medical officer of Medera. And concurrently, he is also the person in charge of the Gene and Cell Therapy Institute at Mass General Brigham, Harvard, overseeing more than 400 professors over there doing gene and cell therapy. And for us, all the 3 decades, we've been really focusing on gene and cell therapy for the heart. And the company is basically trying to commercialize our own technology. And over the next several slides, we're going to tell you what we have done. And Kevin Coster, another co-founder. So let me change my cursor so that you can see better laser pointer. So Kevin Coster, another co-founder, also came from the same background, and he has been more specializing in the human mini heart technology as a discovery engine that I'm going to tell you a little more about. So very quickly, it's a busy slide, but basically 3 take-home messages. How Medera, we believe that is not only unique in cardiac gene therapy, but also in gene therapy in general, and how we are very different from most other gene therapy companies. So 3 take-home messages. One is that we have not seen any side effects. We don't need to use any immunosuppressive therapy like steroid. So no immunosuppressive side effects. We have not seen a single severe adverse event that FDA cares a lot about. And we have not seen any signs of liver damages by us in 21 patients. And also that's been independently validated by Bayer, AskBio, that I'm going to tell you a little bit more about. So, the immediate message is no toxicity or whatsoever before we talk about the efficacy. So the second difference is, instead of doing the conventional systemic IV injection, intravenous injection, we have developed a way to directly inject the drug into the heart as an outpatient procedure. So the procedure is over in about 30 minutes, as opposed to IV injection. The patients need to be hospitalized for about 10 days or so, 7 to 10 days, adding extra cost to the hospital bill. So as such, what is the major advantage is that we can reduce the dosage, the amount of drug that we inject by more than 100-fold. So as such, as I said before, by reducing the dose, we do not see any side effects. And the cost of good is also proportionally lowered. So our cost of good is over the 100-fold. So most companies, their cost of good is about $100,000 to $200,000 US, and for us, it's only about $1,000. So making our drug commercially a lot more viable, because as you must have heard, gene therapy drugs, typically they are outrageously expensive, ranging anything from $1 to $4 million. And for us, when the time comes, we can price very competitively. And the last difference is that most companies focus on rare diseases, though these are diseases that are very rare. And for example, Duchenne muscular dystrophy, maybe only about 50,000 patients in the US. And as opposed to, we are working on a common disease, heart failure, that afflicts 64 million patients. So a big indication, a lot of patients, and therefore the TAM, a total addressable market, is also significantly bigger. So this slide shows you our out licensing record, track record. Roger and I have been in this business for over 25 years, and we used to just license our technology for others to develop to companies like Johnson Johnson, Medtronic, Celadon, and also AskBio Bayer, and and we decided after the Bayer's deal, we decided instead of out licensing for others to develop, we decided to found our own company so that we can make decisions in terms of our clinical trials, our experiments, and also how to commercialize, how to develop our corporate development on our own. Previously, we did not have any say other than licensing the technologies out. So, yeah.
Yeah, could I just jump in?
Yeah.
各位啊，我先提一下，就 Professor Lee 呢是非常少见的，已经把4、5家公司啊或者科技都已经授权出去给这些大药厂了，所以他在 Medera 的个人的投资已经超过3500万美金了。就是这你可以理解这种 commitment 啊，我没有在其他 biotech 公司见到过啊，特别提一下。对 ，OK please。
All right. So for now, so the last row is showing you that we are licensing our mini heart technology to IQVIA, which is our sophisticated investor leading the round. And I'll show you a slide for our clinical assets. We're in the process of talking to multiple pharma companies to basically try to repeat what we had done before, but now with from our own company. So very quickly, since Johnson asked me to compare the 3 companies, meaning Celadon, AskBio Bayer, and also Medera. So very quickly, as I mentioned before, heart failure is a big indication. According to AstraZeneca, there are 64 million patients worldwide, and China alone is about over 30 million, and US and EU combined at about 15 million. So there are 2 major forms. One is called heart failure with reduced ejection fraction, which accounts for 50% of the 64 million patients. And the other form is heart failure with preserved ejection fraction, accounting for the remaining 50%. And this is increasing because of the aging population. Now, actually, HFpEF is more than 50%. And what Roger and I are going to be focusing on over the next 30 minutes or so is going to be primarily about heart failure with preserved ejection fraction HFpEF. And this is our marquee program. This is our #1 program that we are focusing basically the majority of our resources on. I'll tell you why, because this is first in human gene therapy, disease modifying. And also, we are the only company that is working on it. And we will tell you some of the clinical data, ongoing progresses. And so far, we have dosed in our heart failure program over 21. We have dosed so far 21 patients. And as I said earlier, no immunosuppression, no side effects, not a single severe adverse event. That is extremely important through the lens of the FDA. Safety profile has been approved and endorsed. And no T-cell responses. And also no liver damage as constantly monitored. And we have done 21 patients, and that's been independently validated by Bayer, AskBio, about 50 patients or so. I'll tell you what they did and what our relationship is.
Yeah.
So,
Professor, sorry,
yeah.
let me just jump in, right? Because there are some Chinese speakers. 这一页呢，我得特别的解释一下。右上角那边呢是21人的全部使用的已经使用过药物的病人哦。21人为什么这个重要？这个数字它是安全性的一个 number，OK？那用于这个适应症 HFpEF 的只有10个病人，所以美国药监局会同时审批两组，药效性它会审批那10个病人 low dose、high dose。但是呢，会以21人的安全性数据全部看。但我可以特别提一下，在那个 Capital Markets Act 里呢，这也是我们中庸资本的 due diligence 有一页纸呢是专门就提到哪一些是 gene therapy 已经上市的 AAV gene therapy 上市的，它会有6家，全部的安全性都是极差的，很多时候就是肝毒性或者心脏毒性，对不对？ 但是呢，药效勉强能过，但整个药都获批了人数呢也大概跟那个现在21人的安全系数是差不多，在第2或第3期的时候。然后最后一点呢是这样子的，我们公司用的是21人，对不对？那您不要忘记，德国的Bayer right 500亿美金昨天晚上的市值，它左下角那个浅蓝色的 right 就是 AskBio， 就是贝尔的基因治疗的那个 unit，他们已经用了上百人了，也没有安全性的问题所以呢Medera 这里呢是不会跟它竞争，但我们可以随时进入 half-ref， 把另外的50%的市场拿下来，这看上市的情况但待会儿Professor, please talk about the non-compete, right? Whether we can go into the dark blue boxes, okay? Half ref 呢，我们是有机会拿下 half ref 之后再进入 half ref 的这里就能够直接吃掉贝尔的市场。所以您可以理解左下角那块浅蓝的贝尔在买 AskBio 的时候付了40亿美金 ，4 billion US，OK？这个都在你们的那个 Capital Markets Deck 的案例里面。Okay, please, Professor. Thank you.
Okay, so thank you. So my apology because I don't speak Mandarin. I don't understand. I don't speak Mandarin. I'm counting on Johnson to help with the interpretation. So half-ref, heart failure with reduced ejection fraction accounts for the remaining 50%. Three companies are being mentioned here. So Celadon's technology, so about 20 years ago, they focused on, they covered the entire 50% of the half-ref population. So Celadon, I'll tell you more in the next slide as the first generation technology. Second generation technology, Bayer, and they acquired the entire cardiovascular portfolio from us. So, as I mentioned before, after the acquisition, we decided to found our own company to develop third generation technology. The next slide, I'll show you how we are different. And for Bayer, they are focusing on half of the 50% population of HFrEF, meaning 25% of the entire heart failure population. And they're specifically focusing on what we call non-ischemic HFrEF. And for Medera, SLD001, like Celadon did, in that case, in terms of our coverage, we cover the entire HFrEF population, but in order not to have a direct head-to-head competition with Bayer, we are focusing on just the ischemic population instead of non-ischemic that AskBio is doing. Even though AskBio's technology entirely came from us, we're avoiding the head-to-head competition. So we are actually not competing with Bayer by focusing on the ischemic population. So together, combining with the 50% of HFpEF, so you can say the Medera technology covers 75%. And for HFpEF is unique. Nobody is doing this. And 75% of the entire HFpEF, 64 million populations, and for Bayer, the remaining 25%. So this is the basis.
On this point, right? Yeah, I just want to point out, if you just go back one page, right? 所以各位您可以看德国贝尔的进度，或者他们那边如果 proof of concept 他们成了，他们3期临床嘛，左下角那个浅蓝的成了之后 ，Medera 可能都不需要完成自己的3期临床，就会被憋倒了。因为它是个 parallel 的 program， 它的用的科技很类似我们的，然后用的剂量是一样的。Okay, so Professor, how much read across, right, is the Bayer program in phase 3 and phase 2 data, right, 2 hours?
Can you say that again? I'm sorry, Johnson. Can you repeat your question?
Yeah, how much comparable, right, is the Bayer phase 2 and phase 3 data, right, 2 hours? I think you're about to flip to the next page, right? But if Bayer is very successful in that, right, what does it mean for us?
Again, we are not competing. And for HFpEF, Roger is going to be focused on HFpEF. Bayer has no such data, not even one data point, because they are not working on HFpEF. As I said, HFpEF, 50% of the 64 million patients, only Medera is working on it. And for HFrEF, there is some correlation, but also, once again, their data shouldn't be directly compared with ours because they are focusing on non-ischemic patients. We are focusing on ischemic patients. Ischemic patients are usually harder to treat. So, but because they belong to HFrEF, the same category, but the non-ischemic, ischemic are subcategories. So you can kind of like compare, but we shouldn't be really. I mean, they are kind of like apples and oranges in some ways. So just let's be careful with that because, as I said, ischemic patients are harder to treat. Non-ischemic patients are somewhat easier.
Got it. Okay, cool. Let's do the
We
next
can still
section.
make the
Thank you.
comparison. We do have a slide to do that, but it means that we can also share some common findings as well. So
Actually, what I was trying to point out is that the whole field, all the MNC pharma will be looking at Bayer and then at Medera. All eyes on it, right? Yeah. Thank you. Yeah.
So I'll be quick. This is a busy slide. My apology for those of you who are joining the call by iPhones. So, but once again, it describes how the three companies, Celadon, Bayer, and Medera, the different. So Celadon, first generation technology, is started at about over 20 years ago. Some commonalities, we target the same diseases that I mentioned in the previous slide, same molecular pathways. They did not have the mini heart technology that we are using to fine tune. And also, their drugs are very different from how we manufacture our drug these days. Old method, they've got what we call empty capsid, meaning that the vehicles are empty. 70% of the vehicle was empty. Second generation AskBio 30%, and for us 1%. So we have a much more potent and effective drug because of the manufacturing method that we are using 20 years later. And as such, we are able to transduce. The word that we use, transduce, is to make it effective in the heart. And they had a percentage of only 0.5%. And for Bayer and Medera, over 99%. And also the dosages that we are using now are very different from before, let alone the patient selection, patient endpoints are different. So in a way, you can think about like the iPhone or flip phone that you are using now. You can have access to internet. But 25 years ago, the first generation Motorola cell phone, it was analog. You can think about it. It's the same cell phone, but different generations simply have to deal with different manufacturing and engineering technologies. The products we have these days are very different from before. So the cell phones that we have today are very different from the cell phones that we had 25 years ago or 30 years ago. You can think about it. So there's first generation, second generation, and third generation. There's a second generation AskBio, as I've described. The drug is very different. Same method to inject into the heart, except they are injecting manually. For us, we are using, for Medera, we're using an electrical pump. And also the indications, the patient populations are different. And this slide shows you the current competitive landscape, so to speak. Bayer is on the second row, Lexio, Solid, Rocket, Sarepta. These are all basically all cardiac gene therapy companies are publicly listed except for Medera, and we are trying to, change that hopefully in the very near future. So for me to very quickly go through, and only Bayer has access to the intracoronary delivery protocol, meaning the method to directly inject into the heart. And everyone else, they are doing IV intravenous, systemic intravenous, meaning that the dosages are significantly higher. Why? Because we own the IP. And as I said before, Bayer has access to it because they license the entire cardiovascular portfolio from us. And we are in the process of talking to Bayer about reformalizing the license, so they have to pay us because they are now advancing to the randomized phase 2B trial.
Yeah. Can I stop here, right? 各位啊，这张图是可能今天的最重要的两张图之一啊。这里呢是它整个其他在做 AAV gene therapy 的竞争对手。OK， 右手边那个适应症，对不对？你可以看到，其实他们全部做的都是小适应症，什么 DMD 啊，对不对？只有 Medera 和德国的拜耳，就是 AskBio 哈，只是在做最大的心衰的的一个适应症。那你可以看到中间那个有一个图哈，有一个 column 写的是递送 delivery， 对不对？只有 Bayer 和 Medera 是用脚大动脉直接注入心脏的 ，OK， 剩下的全都是用IV 点滴的方式，对不对？然后全身用药 ，OK， 所以呢，他们安全性极差的理由就在这条线上就从第二以下，对不对？那我可以特别提一下，待会儿让教授聊一下 Capricorn， 就是排名第3的那一位为什么 股票翻了5倍，对不对？你可以本来去到200亿美金的，OK，然后它也用的是也是用的 intravenous Professor, just briefly talk about Capricorn and the relationship to you and Professor Hajjar. Yeah.
Capricorn was founded by our Johns Hopkins colleague. They take a very different focus. So we know their program also upside down very well. They are focusing on Duchenne muscular dystrophy, cardiomyopathy. We also have a parallel program that I didn't get a chance to talk about. And uh, uh, Johnson can tell you that that program has a multi-billion potential for Medera. It's what we call a safety program. So we're running it kind of like in the background, but for now we're focusing on HFpEF, which is a significantly bigger indication. But Capricorn, many of their patients who have not reacted to their therapy, they actually are coming to join our program. Their technology and our technology are not mutually exclusive.
Yeah,
And maybe I can. Yeah.
各位 I just want to point out, right? Capricorn 的那个适应症在右手边第3行 DMD-CM, right? 是 DMD 是 Duchenne muscular dystrophy 的病人，但是那个 CM stands for cardiomyopathy， 就是心被压的那个压力，对不对？然后就会死亡。那这个 indication 呢是我们的孤儿药的 indication， 基本上他们已经做成了。就是他的师妹已经做成了，所以读出那个 Phase 3的 data 的时候，股价涨了20亿美金，所以你可以理解，在那个8000万美金的 IQVIA 给 Medera 的 budget 里，6500万是拿下第一行超级大适应症的，1500万只是为了保守一点，对不对？其实可以不用有就是拿下那个 safety line 安全网。那安全网目前市场的估值在12亿到20亿美金之间。Okay, yeah, thank you.
Okay, so you can very quickly to summarize, you can see that Medera has 3 ongoing clinical trials for the heart, and most other companies have only, even AskBio has only 1, Bayer has only 1, and most other companies at most have 2 programs, and they are focusing on rare diseases, and we are focusing on a common disease with 64 million patients. And also in terms of the maturity, we are also more mature. We have completed 2 phase 1/2A trials. One is already, we have completed 2 phase 1/2A trials, and for others, such as Lexio, they're still in phase 1, and Rocket, the market cap has come down quite a bit because they had a patient death. And very similarly, Sarepta from 10 billion down to 2 billion because of patient death and therefore clinical hold issued by the FDA. And I'll tell you why, because primarily have to do with intravenous injection because of the vast amounts of drugs, more than 100-fold that they're putting into the body. Yeah, you can imagine that if you take 2 painkiller, you can treat your headache, but if you take 200 tablets of painkiller, it's going to hurt your liver and something like this. So I'll go over in just a minute. And as I said, everyone else is doing intravenous injection, and we are doing intracoronary, and thereby reducing the dose, eliminating side effects. So this slide very quickly shows you, and this is on the log scale, shows you the dosages that Medera, Bayer, and Celadon were using. Celadon was using they were flying blind. They, at that time, 20 years ago, FDA capped the amount that they can inject. And they didn't know how much to inject, and they were using a very low dosage, thereby getting a 0% efficiency in the heart. And for Bayer and Medera, we are using higher dosages, thereby allowing us to achieve 100% efficiency in the heart, but high enough to induce efficiency, but not enough to induce side effects. As you can see here, and as I mentioned, no side effects, no liver damage, no T-cell responses. But when we go up to a certain point, when I say when you go up to a certain point, like AskBio, you start seeing mild T-cell responses. But compared to Rocket and Sarepta, they are like 100-fold or more than 100-fold higher. As I said, market caps have gone down because of recent patient deaths that they reported as a result of the high dosage and therefore the immunosuppression for rocket and Sarepta liver damage.
Yeah, well, sorry, I just want to convey it in Chinese, right? One page behind, yeah，这页就是最重要的一个 slide 了，OK？刚才我讲2页，我就发你们群里了。右边呢是整个 AAV gene therapy 的这些傻子在做的事情，所以全都是在危险的安全性的边缘上面求这个有效性的。所以你可以看到这边呢，全部的 low dose 的都是用那个 intracoronary 的方式进行的，都是 Medera 有关的公司，要就是 Medera，要就是 AskBio 所以剩下的为什么能做到这个跟刚才我提到的那个 Novel Heart 的那个科技很有关系。待会让教授也解释一下 How do we get the dosing right with the Novel Heart? Yeah, thank you.
So thank you, Johnson. So 2 more slides, and maybe I'll open up for some brief questions before Roger joins. So we've been in active discussion with the pharma companies that I'm showing here, such as Lilly, Johnson & Johnson, Bayer, Astellas, Insmed, AstraZeneca, etc. So, but the plan is that we're going to move on based on the momentum and efficacy data that we have collected, and Roger is going to go over some of you. And we want to go ahead and complete the crossover round to basically complete the next phase of the trial so as to create more value. And the way it goes is that because this is gene therapy and based on the efficacy data, our projected sample size, the size of the trial is going to be significantly smaller than the typical traditional cardiovascular trials that you might have heard about, 100 patients, as opposed to several thousands of patients, and also the time it takes is going to be smaller. Why? It's because when you see a difference of 50% or more, you don't need to do several thousand patients to reach the statistical significance. And Roger will get into the technical data with you. Johnson has asked me to go over the mini heart technology. So basically, we can engineer human hearts, healthy human hearts for screening toxicity, or sick human hearts to screen for efficacy. And the technology has been adopted by companies such as AstraZeneca, Pfizer, Amgen, and it's been also endorsed and accepted by the FDA. And therefore, we have the ongoing trials. That what we can do is that we can use this platform, human hearts, not animal hearts, to discover, to validate the target, and also to optimize the dosages before we put anything into human patients. And this is a technology that is uniquely available to Medera. Celadon did not have it. AskBio did not have it, but they can benefit by working with us. So, and as such, as I said before, it turns out that we are the first cardiology company to have accepted to have received FDA clearances for our IND to launch our human trials based on that, and also to receive the fast track designation. So, and therefore, we're able to accelerate our ongoing HFpEF trial. And the sick heart for HFpEF that we co-developed with AstraZeneca, as I showed in the next slide. So I'm not going to go with the details.
Sorry. Yeah, let me just try to translate this. 我看到教授上来了哈，Professor Hajjar，群里有很多虎妈和学霸，对不对？这个呢就等于是高考前先拿到考卷的情况。他现在 Novel Heart 里面 Mini Heart 连测试了那个药的剂量和安全和这个有效性了，才得上那个 dose。好 ，Yeah, please, thank you.
So thank you, Johnson. And very quickly, this slide shows you the sick HFpEF heart that we co-developed with AstraZeneca. So we have developed the heart, and we also have a unique combination of hardware and software for automated screening. So we are able to show that our sick heart reflects what we see in human patients. And when we put our own drug into this in vitro human mini heart, we're able to reverse the phenotype. And these are the exact same data set that we submitted to the FDA for our IND clearance and also fast track designation that's been awarded to us, and therefore the ongoing clinical trial. So with that, I'm going to stop and happy to take any questions that you might have. And I see that my co-founder, senior colleague, Professor Roger Hajjar has just joined, and he's in Boston. Happy. Um, uh, so I'll let him take over whenever yeah, you guys feel ready.
呀，有没有任何提问呢？因为接下来一个环节我们就直接进入临床数据了。对，好的，
hi everyone. My name is Roger Hajjar, and I'm the co-founder of Medera with Ron. I'm a clinical cardiologist, specialized in heart failure and cardiac transplant. And my work over the 25, 30 years has been really in cardiac gene therapy for heart failure. And in the last 10 years, we've been fortunate enough to bring these programs into Medera and work on 3 different active programs in there. So, Ron, do you have the slides or?
Yes, let me share my screen, switching to a different file. Can you see my slides?
Yeah, I can see the screen.
Okay, so we'll go right into the data set. Maybe we'll start with the intracoronary delivery method.
Maybe, yeah.
So, yeah, and then we get into the patient data.
Yeah.
This is the video.
So this is showing how we deliver our vector, and it's one of the major values at Medera is because we deliver locally to the heart, and we do this through a cardiac catheterization, which is a very simple procedure for us. And we pass a catheter down the coronary arteries and inject the vectors. And you can see here in green, the vectors are being injected and the blood cells. And these vectors contain the genetic material that then goes into the cells, the myocardial cells, and then delivers the gene, the therapeutic gene there, and the therapeutic gene then transforms it into RNA, and from there you have protein that's made, and then the defective protein that's in heart failure is replaced by normal proteins, and then the cells start working better. So that's really the concept in general of how we treat the heart failure patients. Next slide. So we've started one of the trials is in patients with heart failure and preserved ejection fraction, and this is basically half the population of heart failure is based on a heart that's very stiff, so very thick hearts that can't relax. They can contract but can't relax. And that's different from heart failure with reduced ejection fraction, where the hearts are very big and don't contract well. In this case, they just can't relax. And these are patients who have multiple diseases. They usually are diabetic, they have pulmonary lung diseases, they have kidney diseases. And this is some of the profile of these patients that we've treated. And the reason we treated HFpEF is because it's half the population of all of heart failure, but also there's really no treatment for patients with HFpEF. So unlike other types of heart failure, HFpEF really remains a big unmet need. And for us, this was really extremely important. So what this slide shows is that we were able to deliver our vector to these patients. And before we do that, we measure their pressure inside their hearts. We have a catheter that goes in and measure the wedge pressure. This is PCWP in this column. And those wedge pressures are very elevated in these patients. And they are, in fact, what causes the symptoms of heart failure in these patients. So these are patients who are quite sick. And what we were able to see is that in the first dose, we were able to reduce first, they get better by their New York Heart Association. Then we reduce the pressures, and we'll see that a little bit more. We also measured 6-minute walk test and other parameters such as NT-proBNP and KCCQ, which is a quantitative measure of their lifestyle, and they all improve. And the next slide really shows a dramatic improvement in.
Hey, sorry, Professor. On
Yeah.
the previous heat map slide, right? Can you talk
Yeah.
about if there was no intervention, how would it look like clinically, right?
Right. So there's no intervention. This is all would be all red. Most of it at 6 to 12 months, patients really get worse or don't improve at all. So in general, these are patients who continuously get worse and have more and more symptoms. So whether it's KCCQ or New York Heart Association, these patients don't get better, and in fact, most of them get worse. So that's really one of the dramatic changes that we've seen. And the next slide really shows how dramatically we see these changes. We get a mean in changes in the wedge pressure of 30% at rest, and we also do peak exercise. So we exercise them while they have the catheter in place. And these types of reductions correlate with about 47% reduction in mortality. So the wedge pressure is an extremely important parameter for these patients with HFpEF. And we do have improvements in New York Heart Association, their New York Heart Association class, which is another measure of their symptoms, improves by decreasing. We also have a 6-minute walk test that also improved, whereas external controls don't improve, as you can see here. And the mean KCCQ, which is improved at 12 months by 22 points, usually decreases or is not changed. And this is, again, a whole spectrum of disease modification. So it's really important to view this in the context of the fact that when we have these types of changes, these are really monumental improvements that we usually don't see in any types of therapy. And in fact, if you see some of these patients, for example, one patient, as I mentioned, these are multiple diseases, so high cholesterol, hypertension, hypothyroidism, pulmonary hypertension, Raynaud's syndrome, scleroderma, and valvular heart disease. So really just a large number of abnormalities and comorbid illnesses. And this patient went from class 3 to class 2. Their wedge pressure was dramatically improved, 42% decrease, 50% decrease, and then their KCCQ numbers increase. So this is a patient, for example, who no longer meets HFpEF criteria, and HFpEF is a chronic disease, so it's extremely unlikely that you're able to reduce or disease modify in such a fashion in patients who have this type of chronic diseases, and we're able to do that with one delivery of AAV
Yeah.
circuit.
Could you give me 10 seconds to translate this, right?
Yes.
这一页对我来说也是新的一页哈，因为IQVIA 当时把项目转回给 IQVIA 上海的时候，也只不过看到了初步的这个低剂量的数据。那这个病人呢，你可以看到说在低剂量的情况已经reverse 了整个心衰的这个 stage， 对不对？从第3阶段退回到第2阶段了，他只用的是低剂量这挺不可思议的对，好，Professor please.
Thank you. So next slide. So this is showing that how we're able to, by decreasing the wedge pressure and by at rest and by exercise, we're able to improve all-cause mortality or at least correlated with all-cause mortality. And at the same time, these changes in wedge pressure are also there are changes in New York Heart Association and the KCCQ score. So all these things converge together to give you this real disease modification that we see in these improvement parameters. So this would basically correlate very well with mortality, and that's why we feel that it's a very strong surrogate endpoint in these trials, because as Ron mentioned, our goal is to do a trial of 100, 150 patients and not 1,000 patients for the regulators. But when you see these types of decreases in the wedge pressure, this is really what's going to hopefully change that. Now, we also have some data. So we've completed the trial, and all the patients have received the high dose also. And we have early data points for the high dose patients. And what we see is a real decrease, dramatic decreases in these pressures at 6 months, so much earlier than the lower dose. So clearly there's a dose response that we're seeing. It's nice to see. And we feel that this is going to continue at 12 months, and we're going to see some dramatic changes. So what we've seen in the low dose, we're seeing at the high dose, but at much earlier. And one of the investigators feels that these patients, 2 of the 3 patients, do not meet anymore the HFpEF criteria, as we mentioned earlier. And because their wedge pressure has gone back to normal and their heart failure symptoms have reversed. So this is, again, very, very exciting data with the one delivery of AV circa.
Could I just quickly mention, right? One slide behind. Yeah. 去年12月底的时候呢，我们科学家收到了这组数据啊，是从 Duke 的 PI， 因为只有两个这么大的 hospital 能够直接监测他们就 pop champagne 了。我就跟你讲说是，然后在1月的时候呢，1月初的时候去 JP Morgan 的时候，就全部之前聊的那些超级大的基金，像贝莱德Wellington 德诚，其实都是盯着包括 Obiban 就是盯住这组数据。所以刚才我把这组截图了发给你们了。Yes, please, thank you, Roger. Yeah.
Of course. And this is again one of these patients that we're discussing, multiple diseases, 55-year-old woman with anemia, arthritis, bradycardia, Crohn's disease, dyslipidemia, fibromyalgia, nephrolithiasis, so kidney problems, and sciatica. And again, after one delivery of AV circa, she went from class 3 to class 2. And her wedge pressure was dramatically decreased at 6 months, so 60% decreases. So clearly, this patient does not meet any more HFpEF criteria and is really disease modified. And I just cannot be more assertive about the fact that this is really a disease modification that we don't see with chronic treatments in a chronic disease like this. So this is clearly a disease modification because we are really going after the target at the molecular and cellular level, and that's why we're seeing these types of changes.
Professor, could you help us out a bit, right? I want to remind everyone that this is actually the 6 months data. So when these 3 patients go on to 12 months, right, what does yourself or the PI project the wedge pressure to be?
Yeah, I mean, already the wedge pressure is back down to normal. So I think that we expect that these wedge pressures are going to continue to be very low, and we feel that these patients are going to continue to be just very asymptomatic. So they're going to really have no symptoms anymore, for at least from the heart failure perspective. Now, as you can see, these are patients who have so many other disease states. Plus their HFpEF. So if you're able to improve their symptoms from the heart failure perspective, their quality of life is going to be much better.
In a way, an analogy that I always say is you are repairing the engine of your car. Now you have time to also repair the transmission and the exhaust and everything else, but it's the engine that matters the most.
Yeah. Yeah.
Last slide, I think.
Yeah. So this is again for the low dose where we're seeing dramatic changes of 30% or so at rest and an exercise. And these are compared to other treatments, for example, Entresto, which is used for HFpEF, where you see about 10% change, SGL2 inhibitors, where you see 10 to 15% change. And then TX45 is in patients mainly for pulmonary hypertension, and it targets directly the mean pulmonary pressures, and then you see about 20%. So we our just one-time delivery is vastly superior to these chronic treatments that you see here.
Roger, can you comment on like the conventional drugs? The effects are only temporary; they don't last, and how different from our one-time, one-shot approach?
Yeah, so I mean, the gene therapy is a one-time delivery, and it basically gives you this sort of very sustained improvement over multiple years and literally to the life of the patient. Whereas with these drugs, you basically have to take them on a daily basis or sometimes twice a day. And sometimes they have side effects and patients don't tolerate them, so you have to stop and change them to a different type of agent. So, for example, Entresto can reduce blood pressure tremendously, and patients sometimes have to change their doses. SGL2 inhibitors has a lot of side effects, intestinal side effects, and patients sometimes cannot tolerate it. Um, Istaroximine, which is here, has other side effects, and it's again twice time dosing, and it's has side effects that are like digitalis. So, and it's not an
Could
approved
I,
drug, of course.
yeah, could I just jump in because we are all markets people here, right? 右上角的 Entresto 和那个 SGLT2 Farxiga 他们分别是 Novartis 的 和阿斯利康的 number one selling drug，都在100亿美金的销售以上。对，Yeah，thank you。
Even with modest efficacy, and they're not disease modifying. So it tells you how desperate and how big the unmet demand is. Even with modest modification, they are blockbusters, multi-billion dollars of revenue every year.
Right.
So I think it's basically it. Thank you, everyone. So Roger and I are happy to take any questions that you might have. And I see that James Kim has just come on board. James is our Chief Corporate Officer. So James, you want to quickly introduce yourself?
Sure. James Kim, Chief Corporate Officer, responsible for anything BD related and financing/investor related. Before Medera, I was an investment banker for 20 plus years as managing director, focused on covering biotech companies at firms like UBS, Jefferies, and others. Nice to meet you guys.
Thank you, James.
Professor, yeah, maybe I'll start with a question, right? Before I hand over to some of the pros in the chat. Can you give us some qualitative comments from the PIs, from the two centers, right? And why are there only two centers? Can you just name them that in the US that can actually probe for PCWP? Thank you.
Yeah, so the two centers are specialized in these types of measurements. So the reason we have these two centers is because they specialize in these hemodynamic measurements. Again, we're trying to get as high fidelity in terms of the measurements that we take. So it's not just about uh, uh, parameters that are usually used in heart failure, such as 6-minute walk test, New York Heart Association, KCCQ. We needed centers that really do well the PA pressures. And the two centers are Duke University, obviously big medical center, and then UT Southwestern, which is also extremely big medical center in Dallas. So two big medical centers where they have specialized people for these types of measurements. And these are the types of measurements that you can basically defend towards the FDA because they have such good fidelity and give you a very accurate measurements of what's happening with the disease.
Any qualitative comments from the PIs? I saw like an email blurb, right? But please share it with our guys here. Thank you.
Yeah, sure. I mean, we have it on the slides, and the PIs have been extremely uh, uh, shocked at how well these pressures have come down. I mean, they've never seen anything like this, and these are guys who've, centers who do this, day in and day out, and they basically said, these patients do not have HFpEF, don't meet HFpEF criteria, which is very surprising because once you have heart failure, it's a chronic disease and you have it for life. I mean, you can make things better or try to stabilize it, but this is a real a qualitative assessment from the investigators who are the patient's main caretaker. So it's really um, testament to the reversal in the disease or disease modification.
Got it. And my last question, right, is actually what everyone, including Morgan Stanley, cares about. So I'll ask this question in English and in Chinese for the investor there, and then yourself and Ronald, please take a stab at it, right? It's the FDA's attitude or dialogue, right? How likely is it, right, that they look at the combined high and low dose data of our phase 2A, right, and decide whether in April, right, this year, whether that could be a next one, phase 2B/3, could that be a registrational trial, right? What are they going to look at? The safety efficacy profile, what kind of number of patients based on this high dose efficacy, right? Because I'm guessing the high dose is the one that's going into the pivotal, right? Yeah, so I just want to ask your thoughts on this. Just give me 10 seconds to translate this, right? 各位投资人啊，我们特别提到就是今年4月，对不对？FDA 会拿着这完整的 Phase 1B 的数据，然后做一个判断说，咱们下一阶段的100人的这个100到150人的这个Phase 2B， 对不对？/3能不能是注册性临床，对不对？他们会看的是哪一些指标，包括安全性和这个有效性，然后再决定。那当时呢，我们提到的是 Morgan Stanley 讲说其他人的意见都不重要，只要 FDA 看了这组数据，讲说下一个临床就是注册性的，就他们要 sponsor 这个 deal。OK， 好，I'll hand over to both of you for an answer. Thank you.
Yeah. So the FDA, their first concern is always safety, and I think that with our gene therapy product, because it's such a low dose and we're delivering locally, and we've never seen an adverse event, I think they're going to be happy with that. I think that's a really big important criterion for the FDA. The second is that within the heart failure realm, they are accepting now surrogate endpoints. So endpoints that have good correlation with clinical um, effects. So mortality, heart failure hospitalization, and the wedge pressure, as we're measuring it here, has been now shown with multiple different studies that it correlates with mortality, it correlates with the well-being of the patients. So with such a dramatic drop in the wedge pressure, we feel that we have a good case for this to be the next study to be registrational.
May I also supplement? Obviously I agree with everything that Roger said, but I do want to supplement by saying that we are not in a position to say on behalf of the FDA. It's going to be an ongoing process. But what we are seeing here is very dramatic and never been seen in cardiology, HFpEF. So, and considering the unmet demand and these patients, class 3, class 4, the very sick patients, the mortality rate is 75% within 5 years. And there isn't a good drug to modify. And we have shown you some of the improvements of other existing approved blockbuster drugs compared to ours. And given the kind of a difference improvement that we are seeing, so we feel very, very confident that even though the FDA interaction decision-making process is going to be ongoing over the next several years, what we believe is that it's no longer a question about whether this program is going forward or not. It's more like this is a question how this is going to proceed from here for ultimate approval. Keep in mind that this is a first-in-man technology that has not been seen in cardiology. So we expect, and with the RMAT application designation, we will be able to have continuous interaction with the FDA, and therefore in the process, we will learn more and modify this and that. But I think what we are seeing is very, very dramatic. And therefore, there's a high, very high, or even absolute, personally, it's an absolute confidence that this program will go forward. But in the process, we will learn about what FDA expects, and their expectation will be very much data-based, and which is exactly the answers that we are seeking, for the questions that we are asking.
Ronald, sorry, you mentioned the RMAT. For the investors, can you just mention what it is, right? How that is compared to the oncology one, and where are we in the application?
RMAT is Regenerative Medicine Advanced Therapy Designation. It is the equivalent of breakthrough designation, but more specifically for cell and gene therapy. So we had the fast track designation before, at the time or around the time when we got the IND approval to start this trial. So now with the positive data, so RMAT basically is an upgrade once you have completed your phase 1 stats to a trial, and then you ask for that upgrade. And with a 60% improvement in patients that are reactive, and we're showing 34, 36% here because we are, this is this average to some of all the patients, but some are more reactive than others, and this is only 6 months data, right? So, and even that, nobody has seen that before. So we have very, very, very high, extreme confidence that this is, we're going to be getting it. And yeah, so this is my personal view, and Roger, as the world expert, and so please share with the investor group here how you personally feel. But obviously, this is the answer that we're going to be, finding out in the process over the next 2 years or 3 years or so, before we get the ultimate conditional approval. So conditional approval allows commercialization right away, as soon as 2027 to 28. And this going to be followed. We expect this to be followed by confirmatory trial. And we are following up with these patients for up to 5 years. And the confirmatory trial will come after the initial conditional approval for commercialization. And this is the time where we're going to get all the traditional endpoints like hospitalization, mortality rate, etc. But for now, we are using all the co-primary endpoints that are also commonly used in other cardiovascular trials on top of the wedge pressure that we've been talking about, which is a parameter that can be literally measured by a transducer.
OK， 在这里呢，特别提一下哈，就是我们在 QVIA 的判断是 FDA 就今年4月、4、5月的会 accept that 下一个 trial 就是 conditional approval trial 能够注册进临床了。OK， 当然这个也需要这个 RMAT 的助力啊。那我先停一会儿啊，先问一下因为这是已经是临床的最后的问题提问了啊。看看像 Jasper 总Ronald 的 Jasper 总有没有什么问题？对，接下来一环节呢，就是交给 James Kim 总聊一下美国或者 JP Morgan 之后那些大的基金的进度。好了，我先看 Jasper 或者是其他投资人有没有问题。 或 Susie，您可以随时加入啊。对。
Yeah, I mean, as Ron mentioned, the RMAT application is the culmination of all the clinical data that's come forward, and the data is so strong at this point that we feel that this is a very strong RMAT application, and it will allow continuous dialogue with the FDA as we move forward to registrational trial. So this is going to be a pillar in as sort of clinical development program towards a BLA approval for this agent.
And by the way, I want to, we didn't mention that we presented half of these data, not the reversal, because at the time when we made the presentation back in November, we had not seen the reversal yet. And also we did not have the high dose data, but we already presented half of the data at the American Heart Association as a late-breaking clinical trial. And that was a very high profile presentation. We had a lot of attention, and that was November 2025 at the American Heart Association late-breaking clinical trial. And we got a couple of comments from the association about how promising this direction is. And this is an ongoing work, obviously, in progress with very encouraging positive data.
Yeah, so I'll pause for 10 seconds for the Q&A before I hand over to James, right? 可以提问了，谢谢。Okay, I guess there's no questions. Yeah, thank you, Professor Hajjar, right? I know it's late on your side, but please feel free to just drop off. I just want to mention one of our important investors in the group, right, is actually a Harvard student in public health herself, right? So Charlene. So when you guys are back in Boston, we could always link up, right? Yeah.
Please,
but
yeah,
otherwise,
absolutely.
Yeah.
So,
thank you so much.
Yeah, happy to. Thank you.
Yeah. Yeah, I'm just going to ask James to talk about the fundraising process, especially post JP Morgan, right, where everything got accelerated. Let me explain why this arm is blind to myself and IQVIA, is because these are working in parallel, right? We are not reaching out to U.S. investors. Most of our guys are in Asia, right, via Morgan Stanley and Optimus Capital and IQVIA, right? So James, can you just take over and talk about I know there's a long list of names, right? But
Yep.
I just want to focus on the most important, like Dechen, right, which I hold in very high regard. Dechen is part of the group that incubated Grail, right? Their technologies are in there. And we have actually had a couple of co-investments with them, highly respected. In fact, I think they're even better than OrbiMed, right? But in the Hong Kong market, OrbiMed is a lot more active, right? So OrbiMed
Yep.
is coming via IQVIA site, okay? And I know they're in the final stage of IC, right, by end of this month, because of Chinese New Year, actually, and David Wang is not back, okay? But how is Dechen doing? And then the mega funds, right? The BlackRock, Wellington, EQT, and what's that? Ebbingworth, right? I would say that's a bit less, but let's can you just talk about these few? Thank you.
Sure. I mean, I would put the investors in four buckets. So the first one are the high quality healthcare investors that are in final stages of due diligence that should get back to us in the next few weeks. These are all multi-billion-dollar funds, well respected in the US as well as in Asia, that could potentially co-lead with the investors we have already and validate our story to the market. So OrbiMed, Johnson mentioned, Deshan Capital, again, significant presence in Asia and US. They're finalizing their work. B Capital, Sofinova, Vita Capital, and Cormorant is that bucket. The second bucket are large diversified financial institutions and hedge funds that could be meaningful passive investors in the crossover and come in meaningfully on the IPO. So these investors are Aberdeen, which is a $500 billion UK-based investment firm, Marshall Wace, which is a $70 billion hedge fund based in London, Balyasny, $30 billion hedge fund based in Chicago, who could be very big passive investors in this deal and support the IPO. Then there are investors that we presented to in early 2025 that we are re-engaging with the new data and recent corporate updates. Those are funds like Abingworth, BlackRock, EQT, Vivo, and Wellington, who are again high quality funds. And the final bucket is the group that can't come in on the crossover just because their investment strategy, but that like the story and would like to come in on the IPO. These funds are EcoR1, Pivotal, Scion Life Sciences, and others. A very diversified group, all very high quality and large funds that could come in meaningful way on the crossover and the IPO.
And if I were to just focus on a couple of names, right, that we think will be the most beneficial for an explosive Hong Kong IPO. So for yourself and Ronald, right, can you talk about Cheng's commentary, qualitative commentary, and then also on BlackRock and EQT? Thank you.
Yeah, so Desheng, we met a few times, and they actually proactively approached us, especially after our recent high dose cohort data, and in showing our data in context and in relationship to all-cause mortality and heart failure hospitalization. So they proactively reached out to us. They're continuing to pepper us with questions, paying attention where within the next couple of weeks, we should get clarity on their desire to participate in the crossover as it pertains to the amount. The other groups, we mentioned EQT, again, big private equity, private and public life sciences investors. We've met with them 4 or 5 times where the only gating item for them was that their current fund was already maxed out, or they spent all the money in the current fund, where when the new fund opens this quarter, first quarter of this year, they want to really pay attention to us and commit capital in the company. So it's a matter of getting the new fund available to deploy capital and potentially coming in on this deal. Then Wellington.
EQT side. Sorry, EQT side. Are you talking to the life sciences
Yes.
because
yes.
they are different
life
funds,
sciences
right?
team. Yes.
Yeah, because
yes.
I just saw them completing a $200 million round for another biotech yesterday. Okay, cool.
Yeah.
Wellington?
Yeah. And those are all companies. I mean, they did announce recent deals, but those are all companies they have been doing work for 6 to 9 months, right? So it is what it is, but they are very excited about us, and the new fund should be open very soon. So we're looking to get them engaged in this process when that happens.
We would love that OrbiMed comes in this round and then EQT next round with BlackRock with us at Optimus.
Yeah.
By the way, I my first job in London was actually with Marshall Wace in the hedge fund side. Yeah, so,
really?
I know those guys. Yeah. So but cool. And can you talk about Wellington?
Yeah, Wellington, they actually knew about us before we even reached out to them. So they knew about Dr. Hajjar already. They've been following our data and story already. So, that's a conversation that's pretty active, where with our new data, hopefully we can get them on board for this deal. If not, we'll get them on the IPO. But again, it's very good to have a name like Wellington that have been paying attention to us before we even reached out to them.
That's perfect. Last bucket of questions, right, is actually on the corporate side, BD side, right? So can you talk? I only care about 3 names, right? Sorry for being the hedge fund type, right? Because I know that really only J&J, Lilly, and Novartis really matters here, right? Not even Bayer, right? Yeah. Please go ahead. Thank you.
Yeah. So we're talking to big pharmas who seem to like our HFpEF and HFrEF programs and the potential our intracoronary delivery has as it pertains to applying gene therapy for more prevalent cardiac diseases. So these are firms like Amgen, Astellas, J&J, Lilly, Novartis, who are paying close to attention to us, where they're just waiting for some more high-dose cohort data and regulatory clarity around our pivotal trial pathway for HFpEF to really engage there. Then there are European and Asian biotech and pharma companies that are more focused on our orphan cardiac disease program and our DMD cardiomyopathy program. So these are companies like AstraZeneca, Esteve, CKD, which is a large pharmaceutical company based in Korea, Mitsui, Nobel Pharma, NS Pharma, who are more focused on our DMD programs. Then there are some parties who actually like our preclinical pulmonary programs as well. Large biotech firms in the US like Insmed, and some venture capital firms that are looking to license assets to create new biotech companies are evaluating our pulmonary programs. So a lot of recent BD activity.
I think we covered everything today, but I just opened the ground for questions for James, right? It's hard to get him right after JP Morgan, right? So 各位有没有什么问题提问一下给美国的这个融资的这个渠道这边的情况？对。
Hi, I'm Charlotte. Nice to meet all of you, professors and James. I just recently moved from Boston back to Hong Kong, and I just wondering why we are listing in Hong Kong but not listing in Nasdaq in US.
Yeah, we were, I mean, I'm somewhat schizophrenic about this. And as you know, we launched a round in April 2025 in the US. And that was the exact same day when the Trump administration announced the tariff. So I was at a conference with all the other CEO and investors, and many of them, or over half of them, it was a conference organized by Oppenheimer. And more than half of the participants decided just to stay in their room because nobody knew like what to do. But anyway, long story short, I mean, everybody knows that it's been the market, the US market has been very rocky. This sign of discovery, and Hong Kong market is more vibrant. I'm originally from Hong Kong, even though I've lived in the US for like 30 years or so. And I'll take your question, right? So, nobody knows which market is better or worse at any single time point. But what I can tell you from the scientific or medical perspective, and it's all in the public domain, and I've been wanting all these decades, I've been wanting to do precision medicine for Asian. And if we get, there's no win or win. That's not what I'm trying to say, but I'm just saying that from the practical perspective, getting listed in NASDAQ is the way to go. I mean, we can do that. But at the same time, reasonably, you can project that most of the activities are going to be happening in the US, EU. And for now, 95% of our patients are Caucasian. All right. So the focus, the clinical data will be in the US. Listing in Hong Kong, we will continue to do what we've been doing in the US for sure. We're not going to change that. But at the same time, it's going to open up the floodgate or a lot of opportunities, which is exactly what we are now doing already, is that we can also run trials in China. And this is exactly the kind of like regulatory strategy that we are taking, because what we have done in the US, the data will be able to propel what we're going to be doing in China, and IQVIA as the global CRO is going to be helping us not only with the US, but also what we're going to be doing in China. So in China, as you know, they are allowing early commercialization even when you are in phase 2B. So IQVIA is projecting that we'll be able to start commercializing as soon as 2027. So in that case, as far as we've got a good, smart clinical trial design, that we can adopt both in the US, EU, and also in China. So on one hand, we are applying for early approval in China, and the same data set can therefore in turn be used for our US EU trial, multi-center trial anyway, when it comes to pivotal or randomized phase 2B. So the initial plan, as recommended by IQVIA, is that it's going to be about 15% of the size of our next phase of pivotal trial. As I mentioned before, it's going to be roughly, we're projecting about 100 patients, but we are ready to do 150. And still the overall cost in terms of the financial investment and also the time it takes are still going to be significantly less than conventional cardiovascular trials that are of several thousands of patients, right? So 15% of 150, you do the math, is around 20 to 30. So in China, that would be sufficient for us to propel what we want to do in China, and at the same time also facilitating the US FDA track. So we're running parallel tracks, so meaning that we strongly believe, and I hope that you are convinced objectively by the data that we have shown. And as such, one program can facilitate the others. And also, if you take a step back, it is also a strategy to mitigate potential regulatory uncertainties by tapping on both markets. And this is for the first time that we are able to do it. And also, we likely are going to be the first one pursuing this track in cardiology. Not the first in medicine, but first in cardiology. So this is not something that hasn't been done before. And I think Johnson is in a much better position to tell you about what other companies have done and how the market responds to that sort of like dual track efforts as well.
I could add a bit more color, right? Because Professor relies on Optimus for market color and how to go about. 去年最成功的 IPO 是应恩 Duality, right? Duality in November 2024 came to our office, 灰头土脸. They couldn't get US investors to accept $700 million US valuation because two of their lead assets has been licensed out. In the US, they hated that the asset is licensed out because everyone is playing for M&A, the full 10 billion US. 对不对？应恩不合格, right? So 应恩came to Hong Kong and then reassembled another IPO. This is forced by Li Lai, by the way, okay? And then in March, launched at 1.2 billion US and then went up to $6 billion US. Unthinkable if this was a Nasdaq company. 这里有个很重要的一点就是稀缺性在应恩除了科伦博泰以外，对不对？在香港就只有2家真正的 ADC 的 pure play， 对不对？那我为什么要特别跟您讲说你们的 Capital Markets Deck 的第一页 Medera 来上市的时候会有什么标签？就是全部都是 number one in various category， 来第一个全美的 regulatory data 和美国沟通，对不对？第二个是第一家 AAV gene therapy， 然后第三个是第一个cardiovascular pure play 这些 tags 在中国是完全没有的，在美国还有几家 ，OK， 所以这个稀缺性非常难得所以我特别提给你们是讲说他们绝对不应该是去美国 listing， 你可以去 secondary listing there is fine， right？ 不过 primary 一定是由亚洲这边尤其香港来定价，对，当然了在这个基础上呢Optimus 也已经 committed right that 我们会是下一轮只要是 within 1 billion US 我们是 in for 20 million US as a cornerstone， 所以昨天Ronald 呢是见了我们 Millennium 的大 LP 嘛，对不对？其实就是聊这个情况。然后当然了Jasper 总也熟悉博裕资本，对不对？博裕资本的2级市场基金也已经基本上 committed 了。然后我们还没有提剩下的，因为我们现在想聊的是 crossover， 而且是自己人 non-institutional 的完成。因为一旦开启 institutional 就说实话没有咱们什么事了 right that's my point right yeah 所以我为什么会着急着 call 这个 group call 这么短的时间之内，主要的理由是因为 James Kim 那边的进度从 JP Morgan 之后比我们想象的快很多 right yeah so you can understand that once德成 or BlackRock right is coming in this round, the round closes instantly right yeah Okay, any more questions? Obviously, I thought Jasper on as well. Oh, I saw a chat. Okay, no problem. But anyone with questions, right? Especially on the capital market side, this is a good chance. please. Thank you.
Yeah, Ronald and James, this is Jasper. So I'm come from Ronald Therapeutics, which we are focused on the siRNA therapy, and we are also pursuing for the opportunity for the Hong Kong market. So based on what you have already presented, I think I'm very impressed by your technology and what the clinical data you have already demonstrated. So my question would be like, so to be honest, we are also like, so in the last 2 years, we're also considering the opportunity for the NASDAQ. And then we go back from NASDAQ to get a listing on Hong Kong. I think there may be several issues I think maybe you need to consider about. So I think the most important thing is about the liquidity. Like everyone get a listing, we are talking about liquidity, right? But if you look at the Hong Kong market versus NASDAQ, you can see the Hong Kong market, the Hong Kong's liquidity is like only 1 of 10 of NASDAQ, I think. If you look like daily volume transactions of B1, you can see like Hong Kong market only and Asia only like have like 10 to 15% of the total transaction liquidity, but the NASDAQ like dominates for the rest of them. So liquidity will be a quite important issue you need to consider about. But for lots of companies who are getting listing in Hong Kong, because we have some stories in China.
Yeah.
So there is a very important mechanism called Hong Kong Connect. So which can enable the investors in the mainland to buy shares from Hong Kong markets. So that will like to supporting a quite important liquidity. For example, like Johnson just alluded to like Duality. So Duality, it's about like, I think it's about 4 to 5 billion market cap now. And I think one third of its liquidity comes from the mainland funds.
Yeah.
It's not from the international global funds or like Hong Kong funds. So I think this may be, and the question here, I think, is most of the Chinese domestic funds, they do not can have the meeting with you in English. So there may be some difficulties for you to have some fluent communications with them. So I think this may be the most challenge for you guys to get a listing in Hong Kong or have a better market cap management after IPO. So I only have this question for you guys, and the rest of the company, I think it's very fantastic, and I'm looking forward to have any chance to communicate with you personally. Yeah, thank you.
Thank you, Jasper. So nice to meet you.
We have to be really grateful to Jasper. At least my team is, right? Because he is one of the most successful fundraiser. That 3 arms of us couldn't even match one Jasper. In fairness, right? You're very experienced. On your next return trip, right, for OrbiMed, you're in Shanghai a lot with me, right? Please, let's set up a lunch with Jasper and Susie as well, okay? But of course, yeah. Oh, Steven, go ahead. Okay, but I'm just saying that yeah, we will definitely catch up for lunch in Shanghai, because I'm hoping we have more to do with Rona, right? Especially your mini hub platform. But I can imagine the difference, right? So one of the most important conclusion you guys should take away today, right, is that the likes of siRNA, right, the hottest field this year, right, they are more in preventative, right? So preventing HFpEF from even taking hold in stage 1 and stage 2, right? Preventing it, just like Jianfei, right? But what Medera is doing is going from the whole xian, right? Stage 3, 4, a recovery of it. Initially, we're thinking just stabilization, right? But now the drug is looking at disease modifying or even recovery to pre-stage 1 stages, right? So we are approaching it from 2 different arms. So that's what the FDA will consider as well. Okay. So thank you all, right, for your time today, right? You guys are all in a group. For those Hong Kong based, right? Please feel free to visit Ronald in his Novo Heart office, right, in Kwun Tong, right? And he's always here. We can always do lunch and yeah, discuss more. This whole discussion today is being recorded. I'll post it on private on YouTube, so you guys can review this data in more detail, right? Thank you very much, everyone. Thank you. I'll just end off here.
Okay, thank you.
Thank you. Thank you, Johnson.
Thank you. Bye bye.
Bye.
Thank you. Bye-bye.